Hey. All right. Good morning, everyone. We're excited to kick this off. We're going to be doing a talk together, so we'll be just kind of handing off throughout the talk and try to cover some interesting things about the world of research in Huntington's disease. Just first, some disclosures. Just on mine, there are some disclosures that are relevant to some of the companies that are doing some of the work, including Teva and Neurocrine, who are here today, that I do some speaking and consulting for.
And I spend about half my time doing research. So I have funding from the National Institutes of Health, Department of Defense, and a number of companies who are testing compounds in Huntington's disease. They're starred here. I also do some editorial work.
So the outline for today is we're going to start by just talking about some background on some relevant concepts and topics in Huntington's disease, which just to help you understand the research a little bit better. And then we're going to cover the spectrum of observational, symptomatic therapies, and then disease-modifying therapies. So it's a little hard to cover all of the science of Huntington's disease in just a few slides. I'm just going to try to give the relevant concepts that are going to come up as themes in the research that we're going to talk about. And so I apologize if this is basic for some people. This may be more complicated for some people. But the basic understanding about kind of what's happening in Huntington's disease is we know that our DNA is the recipe for making all the proteins in our body, in our brain. Proteins are what our cells need to function. And so the DNA is the message for is the recipe for or the instruction manual for how to make that. And it comes in two strands that have to be unraveled. And then you have a message, which is the RNA, which then gets translated into a protein. And so that's the basics of how proteins are made. And what we know is that the protein's not made normally, or it's mutated in people with Huntington's disease, specifically the Huntington protein. And the result of that, either due to not having the normal protein or due to the abnormal protein that builds up in cells, is that there's damage or loss of neurons in the brain because those proteins aren't functioning properly or are building up. And so the result is that there's atrophy or shrinking of certain areas of the brain that result in different symptoms, which range from different movement symptoms, psychiatric symptoms, and cognitive symptoms, and sort of that combination of problems that progresses over a period of time in people with Huntington's disease. So the other thing we know is we know where the mutation is in that instruction in people with Huntington's. We know where it is in the DNA, and we know that it's due to the message repeating itself too many times. The instructions, which we refer to with letters that are the building blocks of the instructions, C, A, G. We always talk about the repeat length, and that's because we know that the mutation in people with Huntington's disease is that instruction, the C, A, G, in this particular stretch repeats itself too many times. And when it does that, specifically when it does that more than 36 times, sometimes we think about more than 40 times, but when it does it too many times, it makes an abnormal protein, which is shown here with sort of this abnormally long protein in this cartoon. And then that's a mutated Huntington protein. We also know that this is inherited, and so we have two copies of every message for every protein that we make, one that we inherit from each parent. And it only takes one mutated one to cause Huntington's disease. And so if someone has Huntington's disease, typically they have one mutated one and one normal instruction. And that means that they have a 50-50 chance of passing on either their mutated one or their normal one to any of their children. So as we think about research and focusing on treatments, in terms of treatments to actually target the disease process itself, sort of in the big picture, we can target any of these levels. And so there is work being done to target the DNA level, that very first message or that instruction level. Research on targeting DNA is much earlier in terms of where it's at in its phases of development. For now, this is largely in laboratory phases, starting to reach human phases, but we're not going to talk as much about the DNA-targeted therapies because it's not as far along. Where we are going to spend a lot of time today talking about is the RNA or the message targeting, because there is a lot of work being done at that level, trying to stop the message from making the abnormal or the bad protein. And then there are companies that are developing drugs that don't specifically target the the DNA or the RNA, but they protect the cell, or they're aiming to protect the cell from damage or destruction, whether it's due to things like the energy producer, the mitochondria of the cell being damaged, or inflammation in the cell, or some combination of these things. And then the last concept, which are not very simple to wrap your head around, but I'll just try to highlight is we talked about the length, the CAG repeat length, and people often want to know what their repeat length is because it may have some suggestion that the length of the repeat may be associated with the age of onset or the severity of the disease. What we know about the repeat length, that CAG repeat length, is that there is variability of the onset and the severity of the disease with any given repeat length. There is a relationship between the length of the repeat and the age of onset, but there's variability for any number. So what you can see in this graph here is that if someone has 40 repeats, the line kind of on average you can see is kind of close to age 60 for onset of symptoms. If someone has 55 repeats on this graph, it's showing it closer to like 25 at age onset. But there's a lot of bubbles around each spot, which means there's a lot of variability. So for this 40 CAG repeat length, some people develop symptoms at age 80 and some develop them at age like 35. And then that variability kind of goes down a little bit as those numbers get higher. So the repeat length is a strong risk factor for the age of onset of Huntington's disease, but it's not the only factor. And so we're starting to find out that there are other areas of our DNA that code for other proteins that are involved in DNA repair, that when they are mutated may impact the age of onset as either making it later or earlier. And so those are potential targets for some companies working on disease-modifying drugs. And the last concept is this concept of somatic instability, which is that whenever we check for the blood for the mutation, we find numbers around 40, 41, 42. People are born with those mutations at those numbers, but they don't develop symptoms until a certain age. So even though you're born with 40, 41, or 42 repeats, the question comes up is why don't you show symptoms until you're 40 years old or 50 years old? And so what's been found over the last several years is that if you actually look at the neurons in the brain and each individual cell, you'll actually find that every cell has a different number of repeats. We can only really test it in the blood, but if you were to actually be able to test it at every cell's level, some of them might have expanded to 100 or 300 or 500 repeats at any given cell. And that process takes some time to happen. And trying to prevent that from happening is a focus of a lot of companies, is how do we prevent these expansions from happening at every individual cell level. And we think these DNA repair genes that are outside the Huntington gene are responsible for that. So that's going to be just a separate target for some of these treatments. And then finally, just to talk about staging. As you see trials come up, you'll see a lot of inclusion criteria based on what stage of the disease you're in. And so I think just practically when we talk about staging, you'll hear us talk about it in different ways, which can be really confusing. I think it's fine in the clinic, in the day-to-day, to talk about it practically, and we often use things like pre-symptomatic or early or more advanced. When it comes to clinical trials, the staging system that is being used is a little different from that. Right now, this staging system that I have in this picture on the bottom is what is commonly used, and that is that stage 0 is when people have a mutation, but they don't have any signs or symptoms of the disease yet. Stage 1, it's really rare to be able to diagnose people with this at this point in time. Stage 1 is when there are biomarkers of the disease, which we don't typically do in day-to-day clinical practice. This would mean that we find signs of it in the spinal fluid of the mutated Huntington protein, or that we can look at the brain and different volumes of the brain and see that there's some subtle shrinking in certain areas of the brain that we might not see on just a regular MRI. So that's kind of a research level staging. Stage 2 is when people are showing some signs of the disease, but they're not yet impacting their ability to do day-to-day function. So fully independent, but showing some symptoms of the disease. And then stage 3 is when the symptoms are starting to lead to some decline in function. And for that, with function, we typically use this grading scale called the TFC, the total functional capacity. And we rate ability to do things independently, like work, handle finances, do chores, perform day-to-day tasks, be able to be cared for independently at home or in a nursing home. And that's how we define the level of independence. And that's where we'll group people within that stage 3 in terms of the level of functional decline that we see. So these are the sorts of things we look for trials in terms of the staging that we use. But I know that can be confusing. So I'm going to hand it over now to Dr. Hall, who's going to start us off by talking about some of the studies that we have.
So I just want to start with a little bit of a story. So I started seeing Huntington's disease patients almost 25 years ago. And at that time, I was at the University of Colorado, and we had one study, and that study was an observation study. And it was pretty much the first study in Huntington's disease. So I moved to Chicago. And at Rush, I had a good colleague, Kathleen Shannon, who was running the Huntington's program. So she didn't need help at the time. So I went off and did some work in other diseases. And when she moved to the University of Wisconsin, I came back to Huntington's disease. And it had been at least 10 years that I had a little gap. And I could not believe what I saw. I went to the HDSA meeting, and there were all these companies there, and they were interested, and they were doing studies. And I thought, this is something new for us. This is a new era for us. And I think that momentum continues to now. And so some of the slides you're going to hear about today, I hope start the day off for you with hope, because we really need to partner together with all of you to try to get some, medications and something that looks like a cure for folks who have this disease and for your families. So I just wanted to start off with that story because I think it's really a good time for this disease and for all of us. So I want to start just by talking a little bit about research and just some basics about research. So there's two main types of research studies. There are observation studies and experimental studies. So observation studies are when we have a patient in the clinic and we are observing them. We're not giving them a drug. We're not doing anything in particular to intervene in their disease. We're just collecting information. And this is important because we have to know how Huntington's changes over time in order to intervene. So we have to know what we are going to expect for a particular patient with a particular repeat size who has had disease for a certain amount of time. We need to know what to expect because in order to develop a drug to slow it down, we need to have a really good understanding of what we're trying to slow down. Okay, so the observation studies you're going to hear about today, there are some really good ones in Huntington's, and if you're not in them, we're going to encourage you to go into them. On the other hand, we also have experimental studies, and these are the studies you might think about, if you think about somebody testing a drug, that is the type of study that would fall into an experimental study design. So you give a medicine to somebody and you see if their disease gets better. And probably the most rigorous design is if we give a drug to half the patients and a placebo drug to the other half. And a placebo drug is a pill that isn't active, but it looks like the drug that is active. And the reason why we do that, it's required by the FDA for us to do that in order to prove that the drug we're giving is actually changing the disease. So many of the studies you'll hear about in the next half an hour are experimental studies, and we'll kind of talk about them as we go through them. So the first study I'm going to talk about is this observation study called Enroll. How many of you have heard of Enroll? Raise your hands. Oh, how lovely. Okay. So I'm so glad to hear that. So Enroll is the largest observation study for Huntington's disease. There are over 20,000 Huntington's disease people who are gene positive or gene negative who may not know in this study. And it's all over the world. Every site that probably is seeing a lot of Huntington's patients is an Enroll site. And this is an incredibly important study for all of us in the world of Huntington's disease. And the reason why, because this is the study that's teaching us about the disease. And it also allows you to be part of a bigger database that the companies who are wanting to test some of these drugs can look at and say, yes, there is a group of people interested in going into clinical trials. We will come help you. We will develop drugs for you. So it not only helps us learn about the disease, it helps us engage with those companies to work and partner with us. So this particular study, for those of you who raise your hands and maybe you're in Enroll, This study is looking at clinical features. So it's the Huntington's rating scale that we are doing all the time in clinic. Your provider is doing it for you. We're collecting blood samples. We're having you fill out questionnaires. Those are all very validated measures that we use, and it's really important data from year to year. So if you've gone in to enroll, it's been a little while since you've come back for a visit, please come back for a visit because getting that information every year is really important for the study. You can enroll if you don't know your gene status yet. So if you're in the room and you haven't gotten tested, but you're at risk and you want to go in to enroll, you can do that and no one will know what your test result is. I won't know. You won't know. The only people that will know are the people we sent the blood to. Okay, so they don't tell us. So you can still contribute even if you've chosen to wait to test. And as I said, it does serve as a way to get into other studies. So if you're interested in gene therapy, you're interested in some of these experimental studies, going into enroll is really your entry into this. All right, so, okay, I said 20,000, I was close, 28,000, 155 sites, 22 countries. And clinical trial sites are usually based at enroll sites because you've proven to the companies that you have enough patients at that site in order to justify a clinical trial. And the clinical trial are those studies that test medications or gene therapy or other things. The outcomes of this study are to enhance the understanding of Huntington's disease, improve clinical care, and to speed up clinical trials. And I've got this 10-year anniversary cake on here, but actually I think that was a few years ago. So I think we're closer to 15. Okay, so we're closer to 15 years at this point. And I will say that, in fact, they came when we had the 10-year anniversary to Rush, and we did have birthday cake. They brought it with them. So I think they're very proud of this milestone. I'm sure maybe we'll get another one at 15. I'm not sure. Okay, so do you want to talk a little bit about symptomatic? Is that me? Okay, all right, so I'll just keep going. All right, so symptomatic therapies are therapies that we will use in the clinic to treat the symptoms of Huntington's disease. So when we think about Huntington's disease, there's a couple paths we can go. We can treat your symptoms, or we can put you in a clinical trial for a new drug to treat symptoms. And so every patient, it's a very individual conversation with your provider, your physician, to decide whether or not you are going to go on a medicine or whether you don't yet need medicine and go into a clinical trial. Here are some of the medications that we use. Ingrezza, tetrabenazine, and Austedo are three newer medications that are FDA approved for Huntington's disease, and we use them specifically for chorea, those kind of extra movements that you may see. Those, so it kind of looks like people are fidgeting or their upper body is moving. That's what those medications were approved for. So in general, if you have those extra movements, we might be thinking about one of those. However, you can see on the right-hand side of the slide, there are really three big buckets of movements or other issues that you see in Huntington's disease. So in the blue bucket, you see patients can have those extra movements, the chorea. They can have dystonia, which is kind of a twisting movement or a pulling movement, balance problems. They can have eye movement symptoms. They can have Parkinsonian symptoms where they're stiff or they're slow. So every patient looks different. I tell patients, every patient's a snowflake. Every patient looks different than the patient before. And I think the providers in the room would agree that this is the case. And so we have to treat everybody individually. Now, there's more than a movement disorder here. And probably what may be more impactful for patients are the non-movement symptoms. And those are the cognitive symptoms and the psychiatric symptoms. So the cognitive symptoms are things like, problems organizing, just organizing yourself, doing multiple things at once, right? Doing 6 things in a row. Those are skills that may have been normal for someone 5 or 10 years ago, but have gotten harder if they're developing Huntington's disease. It may take longer for people to remember something. It may take longer to do a task because the brain is just functioning a little bit slower. So that's a common symptom that we see. And many times over time, people will develop issues with memory and thinking. All right. So in most patients with Huntington's disease, they'll start to notice some of those symptoms after they have had the disease for a little while. Now, in fact, sometimes patients will come in with that as their initial symptom. And so if that's the case, then obviously we will try to intervene as early as we can in order to try to help patients. And then last, there are psychiatric symptoms. And you're going to hear a little bit more about these after lunch. Dr. Myers, a neuropsychiatrist, is going to talk about these symptoms, and they include things like mood problems, like depression, apathy, just feeling unmotivated, being irritable or agitated. Sometimes patients will hallucinate. So it just really depends on the patient. And so we deal with these symptoms as they come. And it is the reason why we have multidisciplinary clinics in Huntington's disease. So Dr. Beg and I don't sit in a room by ourselves seeing Huntington's patients. If you've come to Center of Excellence, you have seen many folks as part of the team. You see social workers who are here today, physician assistants who are pivotal to helping us. You will see psychiatrist, neuropsychologist, nursing staff, study staff. And the reason why is because of these three buckets. It isn't just one thing that we're trying to treat here. We're trying to treat everything that we can to help your quality of life. And I will say, I've mentioned centers of excellence quite a bit here. Many of you may be connected to one of our clinics. If you're not, and you want to engage with the Enroll study or some of these other studies, now's a great opportunity to get our contact information. You can come in for one of those visits. All right. I think I keep going. Connect. Okay. All right. We split up the slides, and so I just want to make sure I don't keep talking. All right, so Connect is a study that is the most recent study for one of the medicines for Huntington's disease, chorea. And this particular study, some of you in the room may have participated in this study, and I just wanted to give the results back to you today that in fact the study was successful. So this compound is called valbenazine. And the typical dose that we use for patients is 80 milligrams. This was a 12-week study where a portion of the patients took placebo, a portion of the patients took the drug, and it did show that it helped Huntington's chorea. And this is a nice graph on the right that the FDA looked at to approve the drug. So your participation helped get this drug on the market. So you can pat yourself on the back if you were part of Connect. And now everyone can access this from their pharmacy. Okay, now we'll activate. So, okay.
So still on one more symptomatic therapy that I just thought it was interesting. It's a company that it's called Sum, and they used AI actually to find new mechanisms of drugs that already exist. So it's the first time I've really just I thought it was cool because seeing AI actually involved in developing drugs was interesting. And so what they found was a drug that already exists, which is a beta blocker. Beta blockers are often used for high blood pressure, heart issues, that they found that it had properties of a VMAT inhibitor. And the drugs you just heard about, Austedo and Ingrezza, those are VMAT inhibitors that are approved to treat chorea. And so they found that this existing drug had properties on those similar receptors. And so they did do a study in Europe, a phase 2 study. And they showed some improvement in chorea scores compared to placebo. And they're moving forward now. They were concerned about, could this actually negatively impact people's blood pressure, heart rate? The safety looked tolerable. There was some lowering of heart rate or blood pressure. They are wondering, because it also affects other receptors beyond just the VMAT2, which is the one that Ingrezza and Austedo targets, that it also targets a receptor called VMAT1, and that it's also a beta blocker, that there is a potential, hypothetically, for it to also help with mood symptoms. That's what they're looking at. So they're planning a phase 3 study. They reviewed it with the FDA and with the European board recently, and that is something we're expecting to start hopefully in 2026 as another symptom treatment. And then just to think about unmet needs, you heard Dr. Hall talk about all the different symptoms that people will experience, and not everybody's going to experience all of the symptoms, but it differs from person to person. But where are we not addressing things? Where have we not done a good job? I would say cognitive function is one of the big unmet needs in terms of treating people who have symptoms today. And there was a study that unfortunately failed last year. Sage was the company, and it was looking at a drug to improve cognitive performance. And they actually did a study in Parkinson's, in Huntington's, and in Alzheimer's, and all three unfortunately failed. So that ended. There was a study a few years back on irritability. Some of you may have participated in that. And their results haven't moved forward yet. So I think that's a big area of where we need treatments. We have good psychiatric treatments for some things like agitation agitation and mood, but that kind of short fuse that some people get where there's this sudden irritability that can be hard to predict. That's something that I think we need better treatments for. Balance issues in general in movement disorders, we don't have great treatments that focus specifically on balance. Improving chorea, reducing chorea can sometimes help with balance, but in general, balance can be more complicated than just management of chorea. And so I just put what else as like thinking about, I think this is important for us as a community to think about is where should companies be investing their time and efforts is like, what else are the unmet needs for patients and families? And I think this is worth having further discussion about, but just something for you to think about. So switching gears now, a little bit off of symptomatic therapies. This is sort of in an in-between space between symptom therapy and disease therapy. This is a drug called predopidine. The study, Proof HD, was a phase 3 study that was conducted over the last several years. And this is a pill, an oral medication that is focused on stabilizing the cell. So if you remember what I started with, focusing on targeting the DNA or the RNA or the protein, in this case, we're focusing not on any of those targets. We're focusing on the cell itself and trying to protect the cell against damage and destruction and support all the different components of the cell. And that's the goal of this. It's targeting a receptor called sigma-1, and it's thought to help with a lot of those aspects of the cell stability. So in this study, they did not achieve the successful result that they were looking for. They were looking for slowing the progression of the total functional capacity, which is that independence scale, and they wanted to see a difference compared to placebo. And as you can see, their main endpoint on this graph on the left is that the lines for the placebo group and the treatment of predopidine group, they like lie right on top of each other, which means that you're not seeing a significant difference in the decline in functional independence over a period of around 2 years in people who received the drug versus people who received the placebo. So that was considered a failure. However, they did additional analysis and they looked at the people in the trial who were not on any drugs that block their dopamine. So we often use drugs that block dopamine in people with Huntington's, either antipsychotic medications, which help with mood, or VMAT inhibitors, which help with chorea. They both can reduce or block dopamine. And when they looked at the group of people who were not on any of those other medications, they did see some separation in this graph on the right, where the placebo group and the treatment group did actually look like there was a difference, where the group getting the predopidine had a slower decline in their functional independence loss compared to the placebo group. This is a controversial finding in that it wasn't the way they had designed their study, first of all. So the bottom line from their study was that it was safe and well tolerated in terms of actually the side effects in people on the drug weren't very different from the people on placebo. So they tried to get it approved with that data, even though they didn't meet their primary endpoint. And the European Commission that they submitted it to did not approve it. But then, now they're looking at these secondary endpoints that they looked at later on of the group that's not on these anti-dopamine medications. Their thought is that people on some of these other medications that we use in Huntington's, that those medications may have interacted or blocked the effect of the predopidine. And that was their rationale for why some people might see improvement if they're not on these other medications. The argument that you could make, though, is that people on these other medications might have been further along in their disease compared to people not on the medication. And so it's not a given that what they're saying is true. And so they need to study this further. So they're planning another phase 3 study. This is being planned for 2026, Q2, so somewhere in the middle of 2026. And they're planning to look at specifically people who are not on anti-dopaminergic medications. So people who are not on antipsychotics or VMAT inhibitors like Ingrezza or Austedo, and they're going to see if they can reproduce those results on a larger scale in that population. So it's going to be a 1-year long against placebo with an open label extension following people who are on placebo getting switched to the drug, following people over a few years. So this is going to probably start up next year. And then I'm going to start now talking about the Huntingtin lowering. So this is where we start to get into focusing on that message, that RNA that codes for the protein. So we have drugs that are called ASOs, and these are drugs that are delivered through a spinal tap. And companies that you may have heard of that are in this space, Roche and Wave, are two of those companies. And so we're participating in the Generation HD study, which is the Roche study. And this is a drug called tominersen. So this is a treatment delivered through a spinal tap 3 times a year, so every 4 months, in people with early or more subtle symptoms. And so far, it's been going on for a little over a year. The review last April was that there were no safety concerns that would require it to be stopped, so it's ongoing. Initially, there were 2 different doses being looked at, a 60 milligram dose and a 100 milligram dose, and there's a placebo group. They found that some of their data showed that the 100 milligram group was more likely to be beneficial. So they've stopped the 60 milligram group, switched those people to the 100 milligram dose. So now we're just on 100 milligrams or placebo. There are 300 people in this study. Again, early and mild symptoms, and the results are expected next year. So even I, don't know any of the results in terms of how people are doing. And they're also looking at a more selective target because what they're doing is reducing the mutant Huntingtin protein and the normal Huntingtin protein. And we don't really know if that's okay to do, if we need to just lower the mutant Huntingtin protein, or if lowering both is going to be successful in terms of a strategy. So now they're also trying to develop a drug that will target just the mutant Huntingtin protein and not both. So this is more to come in the future. Similar concept is the Wave study. Wave Life has a drug that is also delivered through a spinal tap that has been shown to reduce the Huntingtin protein. In their study, it is selective for the mutant Huntingtin protein. So it doesn't lower the normal one, it only lowers the mutated one. They showed a reduction of about 46% in the mutant Huntingtin protein. And they showed that it was well tolerated. One interesting thing that they showed was there might be a suggestion that the shrinking in the brain, particularly in a part of the brain called the caudate, which is where we know is a specific area of shrinking in people with Huntington's, that they may have showed some slowing of that shrinking in their study, but that has to be confirmed with more people. And so they're getting feedback from the FDA and other regulators about how they want to design their next phase of their study based on their positive results. And I'm going to hand it over again to Dr. Hall.
Okay, so now we're going to talk about lowering Huntingtin protein using viruses. Okay, so who heard about this press release that came out? Yeah, right. Okay, so we're going to talk about that. That's exactly the topic of the next several slides. So uniQure is a company, it's a biotech company, a small biotech company that decided they would get into this field of trying to lower the Huntington's protein. And really the thought is that if you can lower the abnormal Huntingtin protein, maybe it won't clump up in the cells and the cells won't get sick. Okay, so the goal is maybe to try to stop that process. So many of the companies who are working on gene therapy are trying to do that in different ways. And you already heard Dr. Bega tell you what some of those strategies are. So this particular company has used a strategy that they've used in some other nervous system diseases, and they hoped it would be helpful here. So let me tell you a little bit about what they did. So on the upper left-hand side of the slide is what the compound looks like. What it is, AMT-130 is a virus. And it's an adenovirus. And what we do when we're doing these kind of cutting edge gene therapies is we take a virus and we reprogram it. So the virus doesn't make somebody sick. It actually carries a medicine into the nervous system. And this strategy is a very good strategy because we all know viruses replicate, right? They reproduce. And so if you put a virus into the nervous system and it reproduces and it carries a medicine on it, it's going to continue to make that medicine over and over and over and over again. So this is a strategy where you don't just give a pill and you have to give a pill again the next day and you have to give a pill the next day. You actually put the virus in and it may live 10 years, it may live 15 years as far as replicating. And the good news about this virus is it's not going to make you sick because it's programmed to just carry the medicine and not to actually cause disease. So AMT-130 is one of these viruses. So it's an adenovirus that's been used. Again, this type of virus has been used in many diseases, and it carries a gene therapy. And the type of gene therapy is what's called a microRNA. So what it does, this microRNA, is it inserts itself into the Huntington RNA. And when it does it, it prevents it from making that abnormal Huntington protein. And so you can see on the right-hand side this kind of complicated cartoon that shows exactly what's happening. So on the outside of the cell, the far left, you've got the virus, we call it a capsid, that's carrying the gene therapy. It comes into the cell, which is the blue part, and then it goes into the nucleus, where the DNA and the RNA sit. When it gets in there, it works with the machinery of the nucleus, comes back out, and you have this little piece, this microRNA, it attaches to this blue full-length Huntington RNA, stops it from working, and then cuts it into pieces, and it dissolves. Okay, so you can think about this as a therapy that's dissolving the abnormal Huntington protein. Okay, so this is really kind of, one of these strategies. There are many strategies they're trying to use to get the same result. This particular strategy you can't put through the spinal tap. You actually have to put it surgically into the brain. All right, so in the bottom left-hand side, I have this picture. This is a picture, if I cut your head in half and opened it up, that's what it would look like. That's the middle of your brain. This is a surgical procedure where we make a hole in the skull and we take a syringe, we put the syringe down deep into the middle of the brain and we squirt the virus in there, all right? It's in a fluid. And so when we do that, we use a procedure we commonly use in movement disorders. We use this procedure in Parkinson's disease. We use it in tremor patients. We do this procedure standard of care 2 to 3 times a week, every week. But in this case, instead of putting in other electrodes or other therapies, we're putting in this fluid that carries the virus. Okay, so this is a little bit different study because it requires brain surgery. All right, so I'm going to show you a couple slides about the study. So what you can see on the left-hand side are who got to go into this study. And I'll tell you the study is still open, and it's still open at Rush. So if you're interested, please talk to somebody at the table outside at lunch, and we'll put you on the list. So the patients had to have Huntington's disease. They had to actually have symptoms of Huntington's. It's not enough to have a positive gene result. You have to have symptoms. You need to be within the ages of 25 to 65. You need to have kind of medicines that haven't been changed a lot in the last 3 months, and you have to be early. So the patients, many of the patients who got a spot in this study were still working. Okay, so they were working full-time or still working part-time. So they were very early in the disease. And then, as I explained to you, you had to have an area of the brain that this fluid's going into. In fact, it had to be big enough to actually receive the fluid. So if your brain was really tiny and we were going to put the fluid in and it went everywhere, you weren't going to be a good candidate. So about 80% of people who signed up for this study did not qualify because the brain size was not the right size to receive the therapy. Okay, so the most recent 6 patients that were enrolled, they loosened that up a little bit. So the brain could be a little bit different size. So hopefully that'll teach us that we can use a little bit smaller size brain for this. So the patients that we did surgery on over 5 years ago continue to come back and see us. And so those patients we're still doing spinal taps on regularly to look to see if that virus is still working, and we're still collecting data. Now, on the right-hand side of the slide, you can see that this is an American and European study. So the data that came out in the press release was a combination of both. And so what you can see here is that pooled all together at the time this slide was made, we had 29 patients total, and we only have a proportion of patients who have 3 years follow-up. And it's that 3-year data that went to the FDA. Now, what you can see in this gray box here is this sham box. So sham is the placebo. And the FDA required for the first year of this study that there was a group of patients that didn't actually get the drug. But they did go to the operating room and they had a sham surgery. So what that means is that I didn't know when I sent my participants there whether they were getting the real compound or whether they were having a fake surgery. And in fact, it is. They sit in everybody, the surgeon, the operating techs, the nurses, they all sit in the operating room for 6 to 8 hours. and nobody knows. The family doesn't know. Nobody knows whether they got the drug or they didn't get the drug. And the FDA requires this because there's a really big placebo effect, right? Especially in a surgical study. And so they had to control for that, and that was a requirement. All those people who got the sham surgery got to roll over and got dosed. All right. So the study results that were reported were the high dose group in the orange. And what you can see is that, in fact, at 3 years, this compound, this virus, significantly reduced Huntington's disease progression by 75%. And it was on this measure called the Composite Huntington's Disease Rating Scale, which includes the motor signs, a cognitive measure, and function. So all those things put together. And if you could see on the far right, the comparison group was the enroll group. One more plug for enroll. The secondary endpoints were also met. This was the function of the patient. It was also improved from patients who did not receive the therapy. I just want to comment on some of the adverse effects. Most of the adverse effects were around the time related to the surgical procedure and the spinal taps. So we all know when we do spinal taps, there can be a risk of headache, especially afterwards. So there was some of that in these patients. So this slide I made prior to the second press release, but the results that the company put out is that the compound is generally well tolerated. The compound is generally well tolerated, has a pretty good safety profile, and that most of the serious adverse events were right around the time of surgery. And in fact, there haven't been any serious adverse events since 2022. They had planned to go to FDA and then in early 2026 put the drug on the market for everybody. However, that is not going to happen because they had a second meeting, there was a second press release, and FDA would like more data. So what does this mean? It probably means we have to keep going with the study. Now, I will tell you over the last week, many Huntington's disease doctors across the country who were involved in this study put together a letter and sent it to the FDA. And the letter said, please rethink your decision. Okay, so there were multiple people on this email, doctors across the country fighting for the Huntington's disease community. So we don't know yet the outcome of that, but let's hope that FDA takes a serious look at that letter. All right, so we're going to go on to splice modulators.
All right, I know we're running a little over, but this is, I think, another important topic. So I'll try to cover this quickly, but this is targeting the RNA a little differently. And this is with an oral medication now. So we talked about spinal taps, we talked about brain surgery. This is oral. So this is the concept here with splicing modulators is we can introduce a stop reading this abnormal instruction into the reading frame of the RNA of that message. And so there's companies who have developed drugs that can do this. They can actually insert themselves into the message. So when the message is read to make that mutant protein, it starts to read it and it says, stop reading it. And it stops making the abnormal protein, which I think is really cool. So the company was PTC who developed it. Now Novartis bought them. The drug is called Votoplam, and it's a once-a-day pill that they were able to show does, even though it's an oral pill, it does get into the brain. And they were able to show successful lowering of the mutated Huntington protein, and they showed a dose-dependent effect. So what they showed in black line there is the placebo group, and in green is the 5 milligram group, and in blue is the 10 milligram group. And you can see the reduction in the mutant Huntington protein with people who were taking this pill every day for first for a year in a blinded, not knowing if they were getting the drug or placebo, and then over a 2-year period after that. So they showed that it was well tolerated, and they showed additionally trends that it might be favoring towards slowing down the progression on that Huntington's rating scale that we talked about, on the motor scale that we talked about. Although that wasn't the goal of the study, it was to show that it could reduce the Huntington protein levels, which it did. So now Novartis is leading a phase 3 study going forward. It's going to be one dose versus placebo in people with early symptomatic Huntington's disease. So people who are functioning, able to work independently, but who have mild symptoms. And this is going to start next year in 2026. And actually, both Northwestern and Rush are hoping to be sites. There's going to be over 700 people in this study across more than 50 sites, and they're in the site selection process right now. And then a similar concept, another drug that's called company Skyhawk. Their study's called Falcon HD, cool name. They're doing a study in Australia and New Zealand, also an oral drug that is a splicing modulator, introducing that stop codon into the RNA. In addition, though, theirs also targets one of those DNA repair genes, which they're hoping by doing that they can prevent the way those CAG repeats multiply themselves in individual cells. They're hoping that they can target both the Huntington protein and that other gene that is one of the genes that leads to the increase in CAG repeat length. So they had some positive results from an earlier phase study, and they're moving forward into a phase 2/3 study in Australia and New Zealand.
Okay, we are almost done. I know this is a long time to sit through one talk, but I just wanted to add a slide here to show you all the companies working on Huntington's disease. So this is really exciting. So I hope that if nothing else, we send the message at the end of this slide deck that there is hope and that we need you. So we can't do this without partnering with all of you in the clinic, and we will need all of this, everybody here to work together. Have any of you heard of HD Buzz? Okay, I see a lot of people shaking their heads. Okay, so if you don't know about HD Buzz, go ahead and look it up. There are very frequent research updates and articles in there that are really tailored towards patients and families. I've already talked to you about Enroll, so it's just one final plug for Enroll. HD Trial Finder is also on the HDSA website, so you can be part of online studies as well. So here is the information. Take a picture of this slide if you're not already part of our programs. This is how you would sign up for the various studies. Tyler is here, and so you can talk to him directly or one of the study team members, as are several of the other folks on this slide. Emily Zabin's here as well.